My name is Dr. Megan Garland, and it is my pleasure to talk to you all today about my dissertation research titled Determinants of Physical Activity Among Black Women During Pregnancy that was supported by the 2021 Aurora Health MNRS dissertation grant. So, this study examined determinants of physical activity among Black women during pregnancy. And basically, I was looking at two things. I wanted to describe changes in theoretical determinants that could be modified in a future physical activity intervention and the lifestyle physical activity among a sample of all Black pregnant women in the second and third trimesters of pregnancy. And I wanted to examine the effect of these modifiable determinants on changes in lifestyle physical activity while controlling for social determinants of health. I had two measures of physical activity in the study. The first was an objective measure. So basically, participants wore a device that measured their daily activity. And compared to women in previous studies with a mostly Black sample, physical activity was quite low. And there was no change in daily minutes of moderate intensity activity from the second and third trimesters. And this data was collected during the COVID pandemic, so that very well may have contributed to low levels of physical activity. And by self-report, the participants in the study reported walking and dancing were the activities that they engaged in most frequently. I also found that lower pregnancy stress and having given birth previously was associated with higher levels of physical activity. But participants in this sample had very low self-efficacy or confidence to overcome barriers that they experienced to being active, and they rarely received social support from their family or friends to engage in physical activity. So the findings from this study have been disseminated pretty widely. It's been disseminated at CANS and at MNRS. My dissertation was published in ProQuest, and also a manuscript was published in the Journal of Obstetric, Gynecologic, and Neonatal Nursing. I absolutely believe that the study got me my tenure track assistant professorship at Loyola University. And since then, I've secured internal funding at Loyola to further examine the relationships between stress coping and health promoting behaviors among pregnant Black women. And it also supported my submission of an NIH K-23 career development grant that just went in last week. I've been active in MNRS since I joined as a student in 2018. I'm the former co-chair of the Perinatal Sexual and Reproductive Health RIG, an active RIG award reviewer, an active MNRS grant reviewer, and student poster reviewer. And in the future, maybe I'll run for a leadership position. Maybe I will be on one of the planning committees for one of the annual conferences. And I definitely want to mentor junior faculty as I gain more experience. This grant really helped to inform my nursing research trajectory. It provided necessary data to support my K-23 career development grant proposal. It demonstrated that there was a definite need for an intervention specifically to increase physical activity participation amongst pregnant Black women. And it revealed that social determinants of health and stress affect physical activity during pregnancy. And it was really foundational data for my planned adaptation of a tailored walking intervention to improve pregnancy cardiovascular health for Black women. So outside of the grant funding, what has been my most significant experience with MNRS? I learned that nurses are amazing scientists. MNRS is definitely my favorite professional organization. Every conference that I go to, I am just so impressed by the high bar that MNRS sets for nurse scientists, and it inspires me to be a better nurse scientist. I would like to thank MNRS. I truly would not be where I am today without the support that MNRS has provided me in mentorship and in that grant funding to support my dissertation.
From your experience, how do you feel that we can inspire or mentor students to engage in MNRS?
I think that MNRS does a great job with welcoming in students. I think it can be a little bit intimidating. I was intimidated the first time that I went to an MNRS conference, but I was so warmly welcomed, and it really inspired me to become more involved in the organization. As a doctoral student, I felt stretched pretty thin, but there are opportunities within the rigs to contribute in small ways. I started off by just collating and putting together our rig newsletter, and that came out once a quarter. And that was something that I could do. So it wasn't something that was very time intensive. It wasn't something that, where I felt like I was putting myself out there as a scientist because I was just a scared little student. But I got to know the people in my rig. I got to know the amazing things that they were doing. MNRS provides great support as far as like the cost of entry for students into the organization. And that was very helpful for me too. It wasn't a lot of money for me to spend to be part of the organization. And the support within the rigs for like developing into a leadership position where you can be a chair in waiting for a year and learn the ropes a little bit before you assume it yourself. And then you always have the support of past chairs as well. That really makes it accessible and it makes it doable for students who want to become involved in the organization. I'm very sincere when I say that MNRS is my favorite nursing organization. I get more out of my membership from MNRS than I do from some of the other organizations, maybe because it expands the breadth of nursing instead of being a professional organization where, I'm a nurse midwife. And to meet with a bunch of nurse midwives is wonderful and great and stuff like that, but we're all practicing in a lane. And to be able to see how nursing shapes all facets of healthcare is one of the things that's most satisfying and really makes me so grateful to be a nurse.
Hello, I'm Dr. Jamie Morton, the 2021 MNRS Joseph and Jean Buckwalter Dissertation Grant recipient. The purpose of my funded dissertation study was to gain insight into the unique pain-related geographic, socio-environmental, and healthcare system-related factors that contributed to the decision to initiate prescription opioid use and misuse in rural perinatal women. The key findings from my study highlighted suboptimal physical and psychological pain management, past and present traumatic experiences, generational influences, poverty, homelessness, and even food insecurity as contributing to the decision to misuse. Additionally, negative healthcare system experiences such as stigma and punitive threats resulted in a pervasive sense of fear and distrust of anyone in the healthcare system. This ultimately resulted in the perception that these women were forgotten and left behind in the healthcare system that we have. Receiving this grant provided the means to complete my dissertation study, covering recruitment and incentive expenses, which I'm grateful for. Additionally, writing and submitting this grant was an excellent learning experience for me as a NINR T32 predoctoral fellow while I was enrolled at the University of Missouri, Columbia. And it truly inspired me to pursue the path of research. The impactful results from this study really provided insight that I needed further training in pain-related research. This resulted in a completion of a 2-year interdisciplinary NINDS-funded T32 postdoctoral fellowship through the University of Iowa's Carver School of Medicine. To continue advancing my research trajectory, I'm honored to announce I've recently become a faculty member at Sinclair School of Nursing at the University of Missouri, Columbia. While here, I look forward to advancing my program of research focused on chronic pain management with the intent to prevent substance misuse in rural women. Currently, I'm the upcoming 2025-26 co-chair for the Perinatal, Sexual, and Reproductive Health Rig. My future plans include actively participating in conferences, building more collaborative connections with PhD students and new junior researchers that are also MNRS members for future research projects. I also have interest in participating in the MNRS mentorship program and applying for the Leadership Academy in the future. So, to address the question, what currently fuels my fire for research and scholarship? I have this unending curiosity, which has lit a torch of sorts that guides me as I work to unravel unknown elements of a very complex issue and to contribute to meaningful advancements in women's health that will have a real world impact. I want to hold this torch high while preparing the next generation of scholars to continue advancing this research path when I pass that torch on to them. This is going to require active engagement on my part through mentoring future researchers, including them in any of my research studies, so that they can have real-time learning experiences that will hopefully enrich their desire to pursue their own research endeavors.
Hi, my name is Monica Wagner. I'm an assistant professor at Case Western Reserve University, and I received the 2021 M&R Seed Grant to fund the project Pain Omics in the Context of Diminishing Levels of Estrogen. This was a second grant that I have received from MNRS. As a PhD student, I had also received a dissertation grant that was used to help fund the research that provided the basis for this seed grant study. The purpose of our study was to explore gene regulation of women with decreased estrogen in the context of cancer and cancer treatment. Using RNA gene expression and DNA methylation data that we extracted from peripheral blood samples, we first focused on target genes in the adrenergic pathway and then expanded to an unbiased whole genome approach to identify supplemental pathways contributing to chronic pain development, as well as additional novel pathways that may contribute to the development of pain. Participants in the study were postmenopausal women diagnosed with hormone receptor positive stage 1, 2, or 3A breast cancer, scheduled to begin aromatase inhibitor therapy, which is a treatment designed to decrease levels of estrogen and thus decrease the chances of cancer recurrence. Biological samples, demographic, and clinical data were collected at 2 time points. The first was baseline, when all women were pre-estrogen depletion, and the second was after all women had been on 6 months of continuous aromatase inhibitor therapy. Specifically, we explored the hypothesis that 6 months of decreased estrogen leads to changes in gene expression, that was aim 1, and DNA methylation, that was aim 2, and that these changes are related to changes in the pain phenotype of women with breast cancer experiencing decreased estrogen. For each aim, data was generated from blood samples and compared to the pain phenotype of the women at the particular time point. Key findings are that in both gene expression and DNA methylation, we found changes associated with pain. The differentially expressed genes that we found were all active in biological pathways, processes, and molecular networks, helping us to identify biological processes that could be potential targets for pharmacological and non-pharmacological interventions. In the adrenergic pathway, we found DNA methylation sites that might be associated with the development of pain. These changes are especially important when thinking about symptom management and how many symptoms are treated with adrenergic manipulation, both pharmacological and non-pharmacological, including psychological distress, such as anxiety, and physiological stress, such as hypertension. We have presented the results at MNRS, as well as the Oncology Nursing Society Congress and the International Society of Nurses and Genetics. We also used the results as preliminary data for an ongoing study, including expanding our methods to explore cancer and cancer treatment-related pain in head and neck cancer patients. We were also recently just funded by the American Cancer Society Institutional Review Grant to explore changes in DNA methylation related to neighborhood disadvantage and patient-reported pain in head and neck cancer survivors. Thank you.